welcome to part 2 of 3 looking at chapter 8 on photosynthesis. So in part 1 I kind of went over the general process of photosynthesis and how photosynthesis and cellular respiration fit in with the ecology or the environment, or how these things run the biosphere, and why we're kind of looking at these two metabolic processes and how they actually occur. In this section, we're going to start looking at the first reaction in photosynthesis. So here we have this diagram again, and this image here, this is the most important image for this chapter. So remember, this image, it tells you the inputs for the two reactions, which are the light reaction and the Calvin cycle. It also shows you all of the outputs for those two reactions. And then the third thing you want to focus on is where this reaction actually occurs in your chloroplast. So remember, the chloroplast is the organelle that does the photosynthesis process. So this section we're going to be focusing on the light reaction, and then the next part of the chapter we'll look at the Calvin cycle and a few variations in the Calvin cycle that we see within different types of plants. But right now for the light reaction, because it's the light reaction, this is the part where we're going to have the sunlight being absorbed by the different pigments in the chloroplast, and then we're going to convert that energy that's in that light coming from the sun into ATP and NADPH, which is forms of usable energy that the plant can then use to make all of the food that it needs. So in our light reaction, we are going to use light energy, and then in addition to that, we're going to have the water part come in to this. The light reaction takes place in the thiolate membrane, which again are those green stacks that we saw inside of the chloroplast. It's that third membrane. And in this, we're going to produce ATP and NADPH, like I just mentioned. So we're going to basically convert light energy into usable energy in the form of ATP and NADPH. In addition, our water, it's going to turn into oxygen, and then this oxygen is released as a byproduct. We then take that ATP and NADPH and it goes into the Calvin cycle. And here, the Calvin cycle occurs in the stroma, which is just the opening inside of the chloroplast. So we're going to use our ATP and NADPH that was generated during the light reaction, and we're going to use that to incorporate our carbon dioxide and turn it into an organic molecule. So basically, we're going to take carbon dioxide and put hydrogens onto it to turn it into a type of carbohydrate that the plant can then use to pretty much build anything that it needs. So in our light reaction that we're focusing on in this part of the chapter, first we have to talk a little bit about light and how it works. So visible light, it's a type of electromagnetic radiation, and electromagnetic radiation includes everything from X-rays and gamma rays to UV rays, microwaves, radio waves, and also the visible light that we use to see color. Light, it travels as a wave, but it also behaves as particles called photons. And the sun radiates the full electromagnetic radiation spectrum, but we're going to really focus on the visible light, which are basically colors. So the type of radiation that we use to actually see things on Earth. If you look at the middle of this image, we have all of the different types of electromagnetic radiation. Starting on the left-hand side, we have gamma rays, X-rays, UV, the little bit of visible light that we're going to be focusing on. There's the infrared, microwaves, and radio waves. So these are all the different types of radiation that's coming from the sun. And if we specifically look at the visible light, it goes from the smallest wavelength, so the 380 nanometers, which is the purple and blues, all the way up to 740 nanometers, which would be your reds over on the right-hand side. So the really neat thing about, radiation in general, but if you look specifically at the visible light spectrum, So you start off with really short wavelengths at the purple end, and you get to longer wavelengths at the red end. So you have that increasing wavelength from the left to the right. But the amount of energy in that visible light actually decreases as you go from left to right. So as your wavelength increases, the amount of energy in it decreases. So purple and blue actually has more energy in it compared to the oranges and reds, which may seem a little bit strange, but that's how it is. And we're going to look to see what colors plants actually use, so what colors plants get energy from when we look at this full spectrum of visible light. This light energy that's coming down from the sun, specifically that visible light that we just looked at, we have pigments called photosynthetic pigments that are going to absorb that energy that's held in that light that's coming from the sun. And then once that light hits the sun, it hits one of these photosynthetic pigments at the molecular level. That energy is going to be absorbed, and it's going to boost an electron to a higher energy level. So if you look at the diagram on the left-hand side, it says ground state. So here we have an atom, and we have an electron. And remember, electrons, we usually place them closest to the nucleus first, and then as you get more electrons, they go out into the next shells, away from the nucleus. So if we have an electron in the first shell, closest to the nucleus, we have a photon of light that has a bunch of energy. It comes in, gets absorbed, it excites this electron, and it pushes that electron out to the next shell. And this is called the excited state. So we're transferring energy from a photon of light into an excited electron, or a high energy electron. And as you can see, that electron is now sitting in the second shell because it's photoexcited. It's holding a little bit more energy that we're then going to use to do this light reaction. So after that electron is absorbed, it takes in that energy, it gets into that excited state. That electron is very, very unstable. So what happens is the electron, it wants to release that energy as fast as possible. So some of that energy can be released as heat, so it's converted into heat, which is just let off into the atmosphere. It can be converted into light, but what plants do is they take that energy from that excited electron, and that excited electron is transferred to another molecule, or that energy is captured before it can escape as heat or light. So again, we're just transferring energy from one form to another, and we're capturing it so that we can use it, or so that the plant can use it. This whole process, like I mentioned, occurs in these photosynthetic pigments. So most photosynthesizing cells in a plant, they have pigments called chlorophylls and carotenoids. And this image is showing, and hopefully we'll do this in lab, you can take spinach leaves and you do thin layer chromatography on it. So basically you're going to grind up the spinach leaves, put that spinach juice on a paper, and then we're going to run a solvent up the paper, and that solvent is going to separate out all of your different pigments that you can have in that leaf. So there's two types of chlorophyll. We have chlorophyll A and B, which are the green and bluish green pigments. So the xanthophylls and carotenoids, they're the orange and red pigments that we see being separated out on our filter paper on this right-hand side. So here's showing what those different pigments look like at the molecular level. So you can kind of see they're pretty big molecules. Over on the left-hand side, we have chlorophyll A and chlorophyll B. They just have one tiny little difference on them, but they basically have the same structure to them. And again, remember, these pigments, they're responsible for absorbing that solar light and then converting that light into a usable form, or capturing that light. The carotenoids are, here's a carotenoid over on the right-hand side, so you can see that's a pretty big molecule as well. So just to kind of give you an idea of what these different photosynthetic pigments look like. these different pigments that we find in leaves, they absorb different colors in our visible light. So what you can do is you can create what's called an absorption spectrum for each of the pigments. And this just shows which wavelengths are absorbed by your different pigments within the plant. So on this graph, if you look on the x-axis, which is the bottom one, we have our different wavelengths or different colors. So we have violet, which is around 400 nanometer wavelength, all the way over to the red, which is about 650 or 700 nanometers in wavelength. So we have our different colors down at the bottom. On the x-axis, we have a relative absorption of light. So that's just showing how much of the light is being absorbed at that wavelength. And you can see we have chlorophyll A, B, and a carotenoid up here. So if you look at chlorophyll A, which is the red line, chlorophyll A, it has really high absorption around the violets and blues, so you can see a little peak there. And then at the greens and yellows, we have really low absorption. And then it spikes again around the red. Okay. So chlorophyll A absorbs blue, violet colors, and also red colors. Chlorophyll B is very similar, along as well as the carotene. So beta-carotene, except beta-carotene absorbs a little bit into the green wavelength over here. And it does not even absorb any yellow, orange, or red. So you can kind of see what colors these different pigments are absorbing. And if you remember when we looked at that visible light spectrum, remember that the violets and blues, those are the colors that hold a lot of energy. So it makes sense that these pigments are absorbing colors at that end of the spectrum because they have a lot of energy in them. Besides an absorption spectrum, you can also create an action spectrum. This is just looking at the whole plant, and it doesn't break it down by the different types of pigments, but it's looking at the rate of photosynthesis of that whole plant at different wavelengths or at different colors. So as you can see, as a whole, plants generally do more photosynthesis at the purple and blue end, and then at the red end, and then around the greens and yellows, that's where our rate of photosynthesis is the lowest, down here. So to kind of explain this a little bit more, we also have to look at the environment and how plants can change as the environment changes. So the way pigments work, so these chlorophylls, they absorb that red, and then the violet blues that we talked about when we looked at those spectrums. And the green, they don't absorb it, they actually reflect it. And this is why we see leaves as green, because they're reflecting that green wavelength. It comes into our eyes, and then the leaves look green. So generally, plants have a lot of chlorophyll in them. So generally, that chlorophyll is going to cover up the other pigments, the carotenoids, which will appear red and orange in color. So this is why plants look green, and then it kind of relates back to the different types of pigments, and then also how light works. When the environment starts to change, especially around here, even more north of here, so in the fall things cool down, the plants kind of go into the dormant state. This is when plants, they stop producing chlorophyll, because chlorophyll is a really big molecule. It's hard to keep up. It holds a lot of energy. So they stop producing chlorophyll. Some plants will break that chlorophyll down and get energy out of it and store it for the winter. So chlorophyll are no longer produced. This is when we see those other pigments that are kind of hidden during the spring and the summer. And these are going to be your carotenoids. So if you remember, carotenoids, like the beta-carotene, they absorb the violet, the blues, the greens, so those really high energy wavelengths, and they reflect back the yellow, orange, and red colors. So this is why in the fall leaves look yellow, orange, or red, because we're seeing those hidden carotenoids that are always in the leaf, but now we don't have chlorophyll, so these colors are going to show up. Okay, so we have all these photosynthetic pigments that are going to capture that sunlight energy. That sunlight energy excites an electron. That electron is very unstable. It wants to transfer that energy as fast as possible to another molecule, called a carrier. And then this electron, it eventually is going to go down an electron transport chain to produce energy intermediates, so our ATP and NADPH, that we're then going to use in the next step of photosynthesis. So in our chloroplast, we have our light captured in our pigments, and then that energy, or those electrons, they're going to be transferred to photosystems. And there's two photosystems, photosystem 1, or PS1, and photosystem 2, or PS2. And actually, photosystem 2 is the first one in our electron transport chain, and that's because they're named based on when they are discovered and not necessarily in the order that they go. So photosystem 2 is the first one, photosystem 1 is our second one that we're going to see. Okay, so in our cycloid membrane, we have an electron transport chain set up, which is very similar to the electron transport chain we see in cellular respiration. So we have a membrane and we have a bunch of proteins and other organic molecules embedded into that membrane, as we see up here. If we start on our left-hand side, we have PS2, which is photosystem 2. So what happens is that a photon of light comes in, it hits photosystem 2 that has pigments inside of it. That light that's coming in is going to excite an electron, and that electron is going to travel down. So this is the path in red. Our electron is going to travel down from one protein to another in this photosystem. And as your electron travels down, you'll notice that there's hydrogens being pumped into our cycloid membrane, right here. That electron then ends up in photosystem 1, which is more on the right-hand side. It's a light purple. It says P700 on it. Once that electron gets to photosystem 1, we have another photon of light coming in. It excites that electron, and then that electron continues on with the red arrows, and it gets to NADPH reductase. So this electron is going to be caught, and it's used to make our NADPH at the very end on the right-hand side. Okay, so our electron was excited twice. That energy, it's used to either pump hydrogens across into the cycloid membrane, or the energy is used to make the NADPH at the very end of our electron transport chain. This electron, since it's caught at the very end and it's used to make NADPH, we have to replace that electron at the very beginning, and in order to do that, the photosystem 2, it splits apart a molecule of water. So we get an electron from the water, we also get more hydrogen ions inside of our cycloid lumen right here. So we're building up hydrogen ions inside of this cycloid. We have our NADPH created, so the only other thing we need to create is ATP. And we do that through chemiosmosis. So the same chemiosmosis that we've been looking at, we have our hydrogen ion gradient set up inside of the cycloid lumen. Those hydrogen ions want to go from high to low. Since they're ions, in order to do that, they have to pass through ATP synthase, which is going to synthesize your ATP down here. Okay, so we have our NADPH, we have our ATP created, and then the oxygen comes from when we split the water at the very beginning in photosystem 2. So we have our three outputs right here. The inputs would be our light and the water that's coming in. Just to kind of summarize what we went through, so the light is going to excite those pigment molecules in photosystem 2 and photosystem 1. Photosystem 2, that's the first photosystem that was in that membrane. So light hits photosystem 2, it excites an electron, that electron travels down to photosystem 1. Photosystem 2 is also responsible for oxidizing water, or breaking apart water, to generate the oxygen, which is our waste product, and it also generates some hydrogen ions inside of that cycloid. The excited electron is going to release energy in our electron transport chain. that release energy is used to pump more hydrogens into the cycloid to set up our electrochemical gradient that we're going to use for the chemiosmosis to make ATP. Alright, so a lot happens in photosystem 2. Our electron gets to photosystem 1, and then photosystem 1, its primary role is to help make that NADPH. So we take a hydrogen ion, we take that electron, and we capture it to create NADPH. And this helps to create that electrochemical gradient as well that we're setting up, which I'll show you right here. So it's a hydrogen electrochemical gradient that we're going to use for chemiosmosis. Inside of the cycloid, called the cycloid lumen, we get hydrogens increasing from the splitting of water. The hydrogens increase inside the lumen because of that electron transport chain pumping more hydrogens into the lumen. And then on the other side, we're going to use up hydrogen ions to create our NADPH. So we're really interested in creating this electrochemical hydrogen gradient. And then that gradient is going to be used to create our ATP using chemiosmosis. So we use that energy stored in the hydrogen ion electrochemical gradient. Those hydrogen ions want to go through or move across the membrane, and they do that by using ATP synthase, and that synthesizes all of the ATP. This light reaction that we just went through is called a non-cyclic form, electron flow. So this just means that electrons, they begin at photosystem 2 and eventually they end up in NADPH. So they have a non-cyclic, they have a start and an end. So it's a linear process where we produce ATP and NADPH in equal amounts, so 1 ATP, 1 NADPH. However, when we get to the Calvin cycle, we actually need a little bit more ATP in order to run the Calvin cycle. So we have to have a way in order to create a few more ATPs so that the Calvin cycle can actually run. So in order to create a few more ATPs, we do what's called cyclic photophosphorylation. So electrons, instead of starting in photosystem 2 and ending up in NADPH, these electrons, they're going to go back a little bit in our electron transport chain, and they're going to be used to pump a few more hydrogens across into the cycloid lumen. And then the more hydrogens you get pumped across, the more ATP you can synthesize using that enzyme ATP synthesis. So the electrons, when they get to photosystem 1, they're excited by a photon of light. They release energy, they go back a little bit, not quite all the way to the beginning, but they go back a little bit, and eventually they return back to photosystem 1. I'm going to show you that right here. So on the left-hand side, we have photosystem 2, where our electron starts off, it gets excited, it goes down electron transport chain to photosystem 1. In the non-cyclic, that photosystem 1 electron gets excited, it's used to make NADPH. In the cyclic version, here, if we look at our electron, we follow the red arrows. Our electron gets excited in photosystem 1. It flows back almost to the beginning of your electron transport chain, but not quite, and it passes through electron transport again to pump more hydrogens across. And then again, those more hydrogens are used to create more ATP using that chemiosmosis. Okay, so in this section we looked at the light reaction. So again, in the light reaction we needed light and water, and then out of it we get oxygen that's released as a byproduct, and we also get ATP and NADPH, and this all occurs in your cycloid membrane.